Red cells have an average lifespan of 120 days. It is different in different blood type groups because red cells can't sensitize protein. Reparative processes are not possible as red cells age, where anti leads to loss of some of their protein and the activity of some of their essential enzymes decreases. Chemical reactions necessary for the survival of the cells are consequently impaired. As a result, water passes into the aging red cells, transforming its usual discoid shape into a sphere. These spherocytes are inelastic, and as they sluggishly move through the circulation, they are engulfed by phagocytes. Phagocytic cells form a part of the lining of blood cells, particularly in the spleen, liver, and bone marrow. These cells, called macrophages, are constituents of the reticuloendothelial system and are found in the lymph nodes, in the intestinal tract, as free, wandering, and fixed cells. As a group, they have the ability to ingest not only other cells but also many other microscopic particles, including certain dyes and colloids. Within the reticuloendothelial cells, erythrocytes are rapidly destroyed. Protein, we all know the different blood type groups need different quantities, qualities, and types of protein, including that of hemoglobin, which is broken down, and the component amino acids, which are transported through the plasma to be used in the synthesis of new proteins. The iron removed from hemoglobin passes back into the plasma and is transported to the bone marrow, where it may be used in the synthesis of hemoglobin in newly forming red blood cells. Iron, not necessary for this purpose, is stored within reticuloendothelial cells but is available for release and reuse whenever it is required. In the breakdown of red cells, there is no loss to the body of either protein or iron, virtually all of which is conserved and reused. In contrast, the porphyrin ring structure of hemoglobin to which iron was attached undergoes a chemical change that enables its excretion from the body. This reaction converts porphyrin, a red pigment, into bilirubin, a yellow pigment. Bilirubin released from reticuloendothelial cells after the destruction of erythrocytes is conveyed through the plasma to the liver, where it undergoes further changes that prepare for its secretion into the bile. The amount of bilirubin produced and secreted into the bile is determined by the amount of hemoglobin destroyed. When the rate of red cells destruction exceeds the capacity of the liver to handle bilirubin, the yellow pigment accumulates in the blood cells, causing jaundice. Jaundice can also occur if the liver is diseased, like in general, or for example, hepatitis, or if the egress of bile is blocked, for instance, gallstone. The same question, those diseases which are related to different blood type groups